but there's the possibility of many, and they're across the lifespan. And when you think about a biomarker, you have to put it into the context of this diagram. Is it specific for disease? Is it an exposure biomarker? Is it a genetic biomarker? And how does it affect the actual illness state that you're dealing with? A lot of the studies that I'm going to talk to you about now come from a study that I participated in designing when I was at the CDC. It comes from a population-based study of CFS in Kansas, specifically in Wichita, where we followed people over a period of 3 years. After that 3-year follow-up, we then identified a subset of them with CFS, major depressive disorder with melancholic features, insufficient fatigue and symptoms, and insufficient fatigue and symptoms with depression, in addition to non-fatigue. Why is this population important? They came from a population that we have in fact studied over 3 years. So we know a lot about these particular individuals in this subset. These individuals were then invited to enroll in a 2-day in-hospital study that was conducted in Wichita. And there turned out to be 227 individuals altogether who had participated the entire time over that longitudinal study period. We had extensive information on these individuals, very descriptive, self-report, administered information, autonomic nervous system, central nervous system, endocrine immune targets, and genetics. And a lot of this is explained in this particular paper. But my point is, it was a highly characterized population of individuals. A genetic study of those 227 individuals showed that we could target specific genes and look for polymorphisms in these genes, and a subset of those, in this case, the glucocorticoid receptor, tryptophan hydrogenase, and COMT, were all useful in us classifying people with CFS and those without. Now, these did not necessarily distinguish between those with depression and CFS, but they were useful with 76% accuracy in separating CFS from those without. The important thing about these genes is that each one of these genes at one time or another have been implicated in the pathophysiology of CFS. The glucocorticoid receptor is the receptor for cortisol. This is the enzyme that's responsible for serotonin synthesis, and this is the enzyme that's responsible for catecholamine catalysis. So all of these have been implicated in one way or another in pathways important in CFS pathophysiology. So this was a very interesting and important finding. This is another example of how genetic variation in a gene was able to distinguish people with CFS from those that weren't. Focus here on the red area. The more red, the greater the linkage disequilibrium or the association of specific polymorphisms in the glucocorticoid gene with CFS. And this was a significant association of these polymorphisms in the glucocorticoid receptor with CFS and not in those with ISF or non-fatigue. Why are polymorphisms in the glucocorticoid receptor gene important? Glucocorticoid or cortisol is a ubiquitous hormone that affects a number of different functions throughout the body. But importantly, cortisol affects the function of the immune system. It enters into the cell, it binds to the glucocorticoid receptor in a dimeric fashion, it then moves into the nucleus where it regulates the transcription of DNA. Okay? It is responsible for regulating the expression of a number of different genes in the nucleus, all of which affect the function of the immune cell. one of the most consistent findings in the literature in CFS is the fact that people with CFS have immune dysfunction. Another genetic variation was found in the serotonin receptor. And importantly, this serotonin, there was actually three polymorphisms in the serotonin receptor gene HTR2A that were found highly associated with CFS. This particular polymorphism right here was shown to increase the rate of transcription of this particular serotonin receptor gene. And that increased transcription was associated with CFS. How that actually relates to the pathophysiology in CFS is not known. But we do know that serotonin is released into the synapse where it binds to the serotonin receptor so that can then proceed with its necessary functions. And we also know that serotonin itself has a number of different effects, all of which are altered in the body, such as digestion, central nervous system dysfunction. So again, this particular association with serotonin and the serotonin receptor fits plausibly with some of the pathophysiology that we observe in CFS. One last thing on genetics. What we have been able to do with some of these genetic associations is actually apply it back to populations of patients. Again, this 227 patients. Using these genetic markers, we said, can we use this information to subgroup this very heterogeneous population into groups that we might be able to identify, diagnose, and then treat differently than if we just treated them as a whole. And in fact, we were able to identify 5 classes or 5 different subgroups of people with fatigue in this group. 3 of these classes or these groups associated with genetic polymorphisms that also were specific to the types of symptoms and other physiologic markers that that sorted in within these groups. And this was a mixed 227 population. The healthy control group or the non-fatigue group separated out completely clean. So by using combinations of genetic markers, physiologic markers, and symptoms, it is possible to really begin to dissect out the heterogeneity that we see in CFS.
What is class 2?
Right here, this guy.
Oh, down there. Thank you.
We also looked at immune cell gene expression in that group of 227 individuals and combined information, the clinical information that we took, in addition to putting their immune cells onto a chip, an Affy chip, combining that information, and this is a pretty sophisticated analytical approach that results in being able to separate out the people with CFS from those with non-fatigue and ISF. These, again, are the same individuals that you've seen in those prior experiments, but now using a slightly different form of measurement to show that, yes, looking at the immune system in combination with clinical features, people with CFS are distinct. Okay? Digging down a little bit deeper into that information for the gene expression, what that individual was able to do was create a model. In healthy individuals, you have a communication between the immune system and the brain. So this is the classic HPA axis, or hypothalamic pituitary adrenal axis picture, where you have communication back and forth. This has been modified recently to show that the immune system also contributes to this communication network that helps to keep our body in balance. This is our fight or flight system. The immune system is intimately tied into this fight or flight system. Here is balance right here. A healthy individual has immune cells talking back and forth, and we have it balanced. By that previous slide that showed immune gene expression differences, this then changes the model to show that certain cells are now blocked, are unable to signal efficiently to the brain. At the same time, other communication signals are set up where the adrenal is now talking differently to the immune system. The take-home message here is that a very highly integrated and balanced system, which we know exists in people that are healthy, is altered in people with CFS. And it relates to the fact that there seems to be some type of sustained but low-level pro-inflammatory gene expression that's happening in these immune cells, which is consistent with latent viral infection and or latent viral activation. This model is consistent with that. Some more evidence of biomarkers in CFS actually comes from right here in Salt Lake City, the Light team at the University of Utah. These investigators used an exercise challenge model, and they looked at patients with CFS and CFS fibromyalgia patients, asked them to sit on a bicycle, go through an exercise challenge, a moderate level exercise challenge, take blood before and after the exercise at certain times. And the reason for them doing this is they had evidence that certain markers on blood cells that were specific for fatigue or indicative of maybe being fatigue markers, and they had also characterized these markers in the nervous system, and they were specific for pain, so there's certain types of sensory markers, were also expressed on blood cells. So the idea, could we use these in the blood as markers for chronic fatigue syndrome? This work was published last year, and one of their figures actually made it onto the cover of the Journal of Pain. These are all of the markers that they looked at. They had a panel of sensory markers. Again, all of these being measured on blood cells. They had a panel of adrenergic markers, and they had a panel of cytokines, or classic immune markers. This is the result when they looked at these markers on their controls. Baseline, 30 minutes after exercise, 8 hours, 24 hours, and 48 hours after exercise. Not much activity in these markers. They also looked at a disease control population. This is a really important point. Not many investigators do this. They looked for these markers in a population of MS patients. Again, they went through the same exercise challenge, baseline, 48 hours after exercise, not much activity in these markers in the MS patients. When they looked at it in the CFS patients, as soon as 30 minutes after exercise, these markers were highly elevated and increased. This work has been expanded now. This is about 20 CFS patients. It's been expanded up to, I think, 50 or so around there, maybe a little bit more. And these guys are Cindy, Dr. Bateman is actually part of this team too. So this is a very exciting finding, and again, a way to obviously objectify CFS, and it also has implications because we know the function of many of these markers has implications for treatment, okay? So a nice set of potentially promising biomarkers in this population.